Okay, welcome to Module 8 on homeostasis. Remember to review the safety materials and wear goggles when working with chemicals. Read the entire exercise before you begin and take time to organize materials you'll need and set aside a safe workspace so you complete this exercise successfully. Here we're going to investigate homeostatic mechanisms in ID receptors, control centers, and effectors in different scenarios. You'll examine accommodation to temperature by placing hands in bowls of water at different temperatures, and you're going to test heart rate by performing a series of activities and checking your pulse and also checking your body temperature. Upon completion of this lab, you'll be able to define homeostasis and describe the role of homeostasis in complex animals. You'll be able to differentiate between internal and external stimuli, define feedback, both negative and positive, describe the role of receptors, control centers, and effectors, describe two homeostatic mechanisms and relate how each pathway acts to respond to a stimulus, ID some key sensors, control centers, and effectors involved in the maintenance of homeostasis in humans, collect observation on how the human body maintains homeostasis by responding to changes detected in temperature of the external environment, and record heart rate while the body rests, walks, and performs jumping jacks. You'll also create a scatter plot to analyze your heart rate data. The module due dates are shown here: 7/4/16 for the pre-lab survey, 7/8 for the Module 8 lab report, also 7/8 for the Module 8 lab summary, 7/9 for reviewing the pooled class data, 7/9 for the initial homeostasis board post, 7/10 for the homeostasis board response posts. Materials that you'll need are shown here. You'll require ice cubes, a kitchen timer with a second hand or a stopwatch, plastic baggie or plastic wrap, a source of hot and cold tap water. An assistant might be useful, but you don't absolutely have to have one. You can perform these experiments on yourself. From the kit, you'll need 4 plastic cups, 1 Sharpie, and 1 thermometer in a cardboard tube. You'll also need a computer to upload digital camera images, photo editing software such as Word, PowerPoint, or other photo editing software to add labels, leader lines, or text to digital photos, a subject-specific textbook or appropriate reference from lecture content or other suggested resources to get background on what we're doing today. So, what is homeostasis? Well, maintaining a constant internal environment is critical to the survival of complex animals. Homeostasis is the tendency to maintain a state of internal equilibrium or constant conditions. The regulation of internal conditions such as body temperature, blood glucose levels, and pH is possible by the coordinated activity of cells, tissues, and organ systems to maintain a state of equilibrium. An organism's internal environment is regularly changed by external as well as internal stimuli that alter normal body states and functions. What is an external stimulus? Well, an organism's internal environment responds to changes caused by the external environment. As an example, when the human eye is exposed to an onion, tears are produced. The tears help maintain homeostasis by protecting the cells from the external surface of the cornea from irritating chemicals coming from the onion. Internal stimuli also act and are regulated by homeostasis. Metabolic activities are chemical reactions that allow cells to do work. Metabolic activities require a constant supply of substances such as ATP, oxygen, nutrients, salt, and other minerals. The need to replace substances that are used during metabolism is one type of internal stimulus. Cell processes within the cells may also generate products that can act as stimuli. As an example, During exercise, muscle cells use oxygen to do work. The lack of oxygen acts as a stimulus, and breathing increases in order for the cells to maintain homeostasis. When homeostasis fails, in extreme cases, illness or death results. Hypothyroidism, a condition of underactivity of the thyroid gland, can occur for a variety of reasons. The thyroid gland is a mass of cells located in front of the neck below the larynx, and in a healthy human, the thyroid uses iodine to make hormones that regulate metabolism in the body. Sensors in the body signal the thyroid when more hormone is needed, and thus homeostasis is maintained. In extreme cases of malnourishment, insufficient intake of iodine results in a specific type of hypothyroidism, and the thyroid gland does not make necessary levels of thyroxine or T3. The body sensors recognize that more thyroid hormone is needed, and sensors continue to signal the thyroid to make these hormones. However, without the iodine, the appropriate hormones are not made, the thyroid continues to produce incorrect hormones, and the thyroid enlarges, which may result in an abnormally large growth on the neck called a goiter. Hypothyroidism due to lack of nutrition is virtually non-existent in developed countries because iodine is found in most food, primarily, for instance, in table salt. Many medicines have been developed to counteract the homeostatic imbalances that result from conditions such as hypothyroidism. Homeostasis is maintained through a number of homeostatic mechanisms that monitor internal conditions and provide responses to counteract stimuli. Two types of homeostatic mechanisms are negative and positive feedback. In negative feedback, the most common homeostatic mechanism, a response acts to diminish or counteract a stimulus. The function of a home thermostat can serve as a working example of negative feedback. When the air in the house becomes too cool, the thermostat signals the furnace to make heat. When the furnace cuts on, the room becomes warm again. The thermostat signals the furnace to stop producing heat. An example in the human body are sweating and shivering as examples of negative feedback to help regulate internal body temperature. In positive feedback, which is a less common homeostatic mechanism, an amplified response moves the internal conditions further away from the normal set point until we achieve a crescendo event that cuts off the entire positive feedback loop. In the example of the thermostat, positive feedback would occur if the house was too hot and the thermostat signaled the furnace to produce more heat. Only a relatively few examples of positive feedback exist in the human body. One example is childbirth, another is blood clotting, another is vomiting, where a stimulus causes the contents of the stomach to be expelled, moving the body even further from its normal state. Generally, positive feedback mechanisms occur over a discrete, short time span. Okay. The pathways involved in maintaining homeostasis are often complex. In this lab, examples of homeostatic mechanisms have been simplified. Mechanisms rely on three key elements in order to operate: receptors that collect information about the stimulus, a control center that processes information about the stimulus and compares the data to a set point, which is the ideal condition at which the body works, and effectors that initiate activities in response to the stimulus. The figure shown here describes the stimulus, the role of each of the three key elements, and the response. A home thermostat is used as an example. Okay, so you can see here, for instance, we have an area in the home that's got to be maintained at a relatively constant temperature. The home becomes a temperature outside of the set point. A sensor on the thermostat identifies when the home is too cold or warm. The thermostat interprets the change and sends information to the effector, which in this case is the furnace, to either turn on or turn off in order to adjust the temperature back towards normal. The room temperature then returns to the set point until the temperature deviates again and the whole cycle repeats. It's the human body's innate ability to maintain homeostasis that allows us to survive in extreme environments. Consider a human living on the International Space Station, shown here in the figure. The environment has very little gravitational force, and the human body is essentially in perpetual freefall. The human body strives to maintain homeostasis and balance. Living in the space station causes physiological changes to many body systems. In particular, the musculoskeletal system is affected. Whether on Earth or in space, the amount of muscle and bone mass in the body constantly changes as a means of maintaining homeostasis. On Earth, the human body adapts to the weight load that gravity places on the body as a person sits and stands. Tiny changes, including tearing and straining, occur constantly in working muscle. To maintain homeostasis, the body rebuilds the muscle, making them stronger. However, when a person is in space, gravity has less impact, and the body recognizes the decreased weight load, resulting in the breakdown of underused muscle tissue. Thus, in a microgravitational environment, muscle mass reduces. In an attempt to counter the loss, astronauts and cosmonauts exercise 2 hours every day while living on the space station. In this lab, you're going to describe mechanisms that promote homeostasis in the human body, and you'll perform controlled tests of the body's ability to regulate both internal temperature and heartbeat. It's important that the body maintain a constant internal temperature. It's critical because cells may die when they become too hot or too cold. Additionally, the rates of metabolic activity are highly temperature dependent. Homeostasis requires that the human body reflexively avoid environments or objects that are significantly warmer or colder than the core body temperature of 37 degrees centigrade. Skin receptors constantly sense environmental temperature and determine whether the body is gaining or losing thermal energy. Responses to environmental temperatures can be somatic, such as moving into the shade on a hot day, or autonomic, such as sweating to induce evaporative cooling. Your heart rate is also a variable that acts to maintain homeostasis. The cardiovascular system transports oxygen, vital nutrients, sugars, and minerals to the body's cells. The volume of blood that the heart pumps changes to accommodate the needs of the cell. When a person exercises, blood flow has to increase to accommodate the working muscle cells. Thus, heart rate increases in response. However, there is an upper threshold for the number of beats that can occur over a given time. Because the heart requires time to expel the blood, it holds and refill the blood from the body. So in this exercise, you'll identify positive and negative feedback and determine the stimulus, receptor, control center, effector, and response for various stimuli. You'll describe how each form of regulation contributes to maintaining homeostasis in the body. So stay tuned for the experimental procedure. Okay, welcome back. This podcast will deal with the procedures involved in the homeostasis lab. Relatively simple experimental setup here. What you're going to need are ice cubes. I've got some crushed ice here. Okay, a kitchen timer or a watch with a second hand, a partner, but that's not absolutely necessary, plastic baggies, a source of hot and cold water, which I have here in the room, 4 plastic cups, a Sharpie, and a thermometer in a cardboard tube, and as always, a computer to upload digital camera images, editing software like Word or PowerPoint, to add labels, leader lines, or text to digital photos, and of course the information from the lecture component of the course. Okay? And our first activity is going to be positive and negative feedback. So what you have here are a list of scenarios, okay? So scenario 1, the body is too hot. Temperature in the body is sensed by the hypothalamus, an area of the brain that regulates hormones that address not only temperature, but hunger, mood and sleep, and sex drive. When a person becomes too hot, they begin to sweat, which reduces the temperature of the body through evaporative cooling. The blood vessels in the skin dilate so that warm blood is closer to the surface of the skin. Okay? And so that'll be our first scenario. All right? Scenario number 2, the body is too cold. When a person becomes too cold, the blood vessels in the skin constrict to conserve heat. A person may begin to shiver, which is an involuntary contraction of muscles that generates heat and raises the overall body temperature. Scenario 3, blood glucose levels go up. Cells of the pancreas, which is an organ that's wedged between the small intestine and the spleen and behind the stomach, determine the set point for blood glucose. After a meal, glucose levels are elevated and the pancreas detects this change and makes insulin. Insulin and glucose move into the bloodstream where they're transported all over the body. The insulin allows glucose to move from the blood into the muscle and the liver, where the glucose is stored or used as energy for work. Scenario 4, blood glucose levels drop. When glucose levels are low, the pancreas produces glucagon, which signals the release of stored glucose. And in the final scenario, scenario 5, the body's epithelium sustains an injury. When the body sustains an injury, such as a cut, scrape, or burn, a message is sent to the brain that the injury has occurred. As a result of the injury, blood vessels break and blood flows into the wound. Proteins and platelets in the blood help to form a clot, which is a temporary seal between the area of the injury and the inside of the body. This will eventually form a scab, and epithelial cells will be regenerated after the dead tissue is removed by the cells that repair tissue and fight pathogens. Those are components such as fibroblasts, which are found in connective tissue, epithelial cells, which regenerate the upper layer of the skin, and white blood cells, which fight pathogens. So, part 2 here, record the type of feedback mechanism for each scenario in data table 1 of your lab report system, using, again, the information previously addressed in the podcast. Determine the stimulus, the receptor, the control center, the effector, and the response for each scenario, and record your findings in data table 1 for each stimulus. List whether the source is environmental or metabolic. Okay? And then final step, describe how each scenario contributes to homeostasis in the body. Identify why maintaining the internal system this way is important to survival. Okay? And then make sure to answer the questions on page 9. All right? So that's exercise 1. All right, now I'll join you in just a moment and we'll deal with exercise 2. Okay, in exercise 2, you'll test the body's sensitivity to temperature through exposure to a series of water baths and various temperatures. Stay tuned for these exercises. Okay, welcome back. We're now on to activity 2. What you'll need for activity 2 is some hot water. I microwave some in a coffee mug. You can use hot tap water if you like. And I used ice, and then I used regular tap water. And I kind of play around with it to get the right temperatures. 10 degrees, room temperature, which is 20 degrees C. 30 degrees C, okay, which is about, I would say, less than a third of a cup of really hot water and the rest of just room temperature tap. And then 45 degrees C, which is about 2/3 of really hot water and 1/3 of regular tap, okay? So once you've got your cups and you've recorded your temperatures on your thermometer, entered that into your lab report, Next thing you want to do is locate your kitchen timer. Okay, so we turn this sucker on, and you want to dry your hands, make sure they're not wet, and with your timer, place one index finger in the 45-degree water and the other in the 20-degree water. And again, look at figure 3 for reference, okay? And allow your fingers to remain in the cups for 30 seconds. Then place both fingers in the 30-degree water for 30 seconds and note the sensation in each finger, okay? So 1 in 20, 1 in 45, 30 seconds, then into 30 for 30 seconds. All right, so we'll start. Okay, now switch. Okay, go to 60. Again, note the sensation in each finger. Okay, stop. Record your observations, okay, in your lab report and data table 2 of your lab report assistant. Now place your index finger in the 45-degree water and the other in the 30-degree water. Allow them to remain there for 30 seconds, then place both in the 10-degree water for 30 seconds and note the sensation in each finger, okay? So we're going to reset, okay? So we're going 45 and 30 and then into 10, right? So we will begin. Okay. Record your observations. Dry your hands. Okay. Enter your data for your second trial in data table 2. Describe the sensation when you moved your finger from 45 to 10 and the other finger from 30 to 10. Okay. And then you want to answer the questions that are on pages 10 and 11. All right. Okay. Our next. In this exercise, you'll collect data on heart rate doing a series of exercises. So you're going to do jumping jacks and you're going to walk in a circle rapidly in order to have an effect on your body temperature and your heart rate. Again, the caveat here, if you have any concerns about physical activity, consult your physician and get approval before beginning this exercise. If you're not able to do these activities, try getting a partner. So we start by locating a kitchen timer stopwatch with a second hand. Sit comfortably at rest for 30 seconds and determine how many times your heart beats in 15 seconds. Record the number of beats as resting beats per 15 seconds under trial 1 and data table 3 of your lab report assistant. Now, how do you determine that? Well, you can take your carotid pulse here or your radial pulse here. Next, continue to rest for 30 seconds. Again, determine how many times your heart beats in a 15-second interval and record the number of beats as resting beats per 15 seconds under trial 2 in data table 3. Repeat step 2, resting and recording the number of heartbeats until 10 trials have been recorded in data table 3. And again, why do we do this? To make sure that any variation or any number that you get is reliable and not due to variation from random chance. Then calculate the number of heartbeats per minute and record as resting beats per minute for each trial in data table 3. To calculate beats per minute, remember to multiply resting beats per 15 seconds times 4, because there are 4 15-second intervals in a minute. Next, place a thermometer in a plastic baggie or piece of plastic wrap. Place the thermometer under your tongue for about 1 minute and record your temperature in data table 4 of your lab report assistant. Then we'll start with your first exercise regimen. We'll do walking trials. Start by resting while sitting for 2 minutes. Walk at a quick pace in a circle for 30 seconds. Stop walking and determine how many times your heart beats in 15 seconds. Record the number of beats as walking 15 beats per second under trial 1 in data table 3. Repeat steps 6 through 8, walking and recording the number of beats until 10 trials have been recorded in data table 3. Calculate the number of heartbeats per minute and record as walking beats per 1 minute for each trial in data table 3. Again, determine your body temperature by placing the covered thermometer under your tongue for about 1 minute. Record your temperature in data table 4. Next, with jumping trials, you want to rest while sitting for 2 minutes, then do jumping jacks for 30 seconds. Stop jumping and determine how many times your heart beats in 15 seconds. Record the number of beats as jumping beats per 15 seconds under trial 1 in data table 3. Repeat steps 12 through 14, jumping and recording the number of beats until 10 trials have been recorded in data table 3. Calculate the number of heartbeats per minute and record as walking beats per 1 minute for each trial in data table 3. Again, determine your body temperature by placing the covered thermometer under your tongue for about 1 minute. Record your temperature in data table 4. Rest while sitting for 2 minutes. Create a scatter plot for the number of beats per minute versus trial number. Plot the trial number on the independent axis, which is the x-axis, the horizontal one, and the number of beats per minute on the y-axis, which is the vertical one. Create 3 lines to represent the heart rate while resting, walking, and jumping. Insert the graph into graph 1 of your lab report assistant. An example is shown here in the panel. Okay, so we've got our resting numbers here, our walking numbers here, our jumping numbers here. So again, try and anticipate what the effect of each of these states is going to have on your pulse rate and on your body temperature, and then see if your data support your hypothesis. If they do, your hypothesis is likely correct, and you can further test your hypothesis by increasing the amount of exercise and seeing if the heart rate and the temperature continue to go up. If your results do not support your hypothesis, you'll have to form another hypothesis and test it. Okay? So, stay tuned for a demonstration of these jumping trials, and once that's completed, make sure to carry out your experiments and generate your lab report, and I will see everybody in the next video podcast. Thanks for listening. Experiment is going to be exercise 3, homeostasis and heart rate. Again, what will you need? A kitchen timer or a watch with a second hand. Sit at rest for 30 seconds. Determine how many times your heart beats over 15 seconds. Record the number of beats as resting beats per 15 seconds under trial 1 in data table 3 of your lab report assistant. Continue to rest for 30 seconds. Again, determine how many times your heart beats over 15 seconds. Record the number of beats as resting beats per 15 seconds in trial number 2. Repeat step 2, recording the number of heartbeats until 10 trials have been recorded in data table 3. Okay? And the reason we do this, repeated trials, is to make sure that any variation we see is not due to random chance, but is in fact a number that we can trust. Okay? So you might have, for instance, you've taken your pulse and you might be agitated at some point, and at another point when you're taking your pulse, you might be more relaxed. And that's going to have an effect on your resting rate. So by taking 10 trials, that averages out, and we get a number that we have more confidence in because it's more likely to be statistically significant. Okay? So calculate the number of heartbeats per minute and record this as resting beats per minute for each trial in data table 3. To calculate beats per minute, multiply resting beats per 15 seconds times 4. Now figure 4 on page 12 shows you a couple of places where you can take your pulse. I like the neck because it's easier to feel, but some people are more comfortable with radial pulse, which is on your wrist, on your thumb side. But again, let's just demonstrate. Go ahead and you want to reset your timer, okay? Find your neck pulse, okay? Like this. And then we're going to start it and go 15 seconds and count the number of beats. Okay, so on that trial, I got 20 beats in 15 seconds. And so I would repeat this total of 10 trials, and then we're going to record all of those trials in your data table 3. And then the next thing you're going to do is move on to temperature, okay? You're going to place a thermometer in a plastic bag or a piece of plastic wrap and place the thermometer under your tongue for about a minute and record the temperature in data table 4 of your lab report assistant. Okay, so relatively straightforward. Same thermometer you've been using for the other trials. The plastic bag is designed again to keep you from putting something nasty in your mouth. Okay, so it's a nice clean plastic bag. Okay, it's a good idea to note the initial temperature on the thermometer, which is 20 degrees, which is room temperature in here. Place the thermometer in a baggie. Okay, and then reset your timer, and then place it under your tongue for 1 minute. Okay. Temperature I got was about 30 degrees. Okay. Typical human body temperature is around 37. Why would the temperature be lower? You're working through a baggie, okay, and that's going to have an effect on the sensitivity of the thermometer. Whereas with a standard thermometer that rests right against the skin, the reading is going to be a little bit more accurate, okay? So you record that in data table 4, okay? Next thing you want to do are the walking trials, okay? So rest for 2 minutes, then walk at a quick pace in a circle for 30 seconds. Stop walking and determine how many times your heart beats over 15 seconds. Record the number of beats as walking beats for 15 seconds under trial 1 in data table 3. Repeat steps 6 through 8, recording the number of beats until you've done 10 trials that have been recorded in data table 3. Calculate the number of heartbeats per minute and record as walking beats per 1 minute for each trial. Determine your body temperature by placing the covered thermometer under your tongue, just like we did for the resting rate, and record your temperature in data table 4, okay? So, walking in a quick pace in a circle for 30 seconds. Relatively straightforward, right? I would get a chair out, I would clear some space. Again, you don't want to trip and fall. Okay? You want to have enough room to walk, and you're going to do this for 30 seconds, so okay. And sit. Take your radial pulse. Okay? And record that number. Okay? And then turn around and take your temperature. Again, note the initial temperature on the thermometer. Okay, record the temperature. I'm about 5 degrees higher than I was at resting on this trial. Again, you're going to want to do this 10 times. Enter the data in data table 4. Okay? All right. Clear your timer. Next, jumping trials. Rest for 2 minutes. Okay? Then do jumping jacks for 30 seconds. And then you're going to get your pulse over 15 seconds. Enter that in data table 3. Call that jumping beats per 15 seconds. Repeat this until you've got 10 trials, and then take your temperature with the thermometer and note it in data table 4. Okay? So again, for those of you who don't know what a jumping jack is, I can demonstrate. Okay? Jumping jacks for 30 seconds. So we will start. Okay, stop. Take your radial pulse. Okay. Now, on that trial, I was about double what I was at resting. Temperature. Again, note the initial temperature. Okay. All right. And that's about, on me, 5 degrees higher than resting. Again, you're going to want to repeat this for 10 trials. And then you're going to generate a scatter plot using the information from your resting, your walking, and your jumping trials that will plot the number of beats per minute versus the trial number. Plot the trial number on the x-axis and the number of beats per minute on the y-axis. Generate 3 lines to represent the heart rate while resting, walking, and jumping, and insert the graph into graph 1 of your lab report assistant. An example graph is shown in figure 5. Okay, so if you look on page 14, you'll see an example of some sample data graphed with the 10 trials and how it should look. Again, answer the questions on page 14. That's going to be part of your lab report, and that draws the exercises in the homeostasis lab to an end. Now, what's the point of this lab? The point of this lab is to understand that in order for the human body to function normally and stay out of a disease state, there are certain values that have to be maintained within a tight window. Okay? For instance, your body temperature, your body fluid content, your body electrolyte content, your heart rate, your respiratory rate, your blood oxygen and CO2 levels, just to name a few things, cannot get higher than a ceiling value or lower than a floor value, because if they do, you'll have a disease state. The body won't operate normally. If they get too far outside of this window, then you're going to have death. So the body will do anything it can to try and keep you within those particular ranges. Okay, and we gave you some examples. The example of body temperature is a good one, right? If your body temperature begins to rise for whatever reason and continues to rise beyond the upper limit, what will happen is that the increase in body temperature will affect chemical reactions in the body. The proteins that help chemical reactions work, which are called enzymes, will begin to denature. The result of that is that those chemical reactions will stop. If those chemical reactions stop, the cells can't make the energy they need to keep living. Other chemical reactions that support life will shut down and the cells will die. As the cells die, the tissues die. As the tissues die, the organs die. As the organs die, the organ systems shut down. Okay, so that's one example. Flip side, body temperature gets too cold. What happens to chemical reactions as they cool down is they slow. There's a rule of thumb that says that for every 10-degree drop in chemical reaction temperature, the rate of the reaction slows by twofold. Well, if you can see, if the temperature keeps dropping and dropping and dropping, eventually the reaction will become so slow that the cell won't be able to meet its metabolic needs. It won't have enough energy to keep going as a living thing. And the cell will die, and the same process will continue, right? The tissues will die, the organs will die, the organ systems will shut down, and the organism will die. So the body does everything it can to try and maintain those values within a range that's compatible with life. And that's what we're demonstrating with these exercises today, which assess things like heart rate and temperature, which are just a couple of the values that the body strives to maintain within a nice tight window. Okay? So with that, we